Good day everyone, this is Aljan PD from LO15 class, and today we'll be having a discussion about the cell cycle, focusing on mitosis and meiosis under stages. But before anything else, let's define cell cycle. So what is cell cycle? So cell cycle refers to the ordered sequence of events that occur in a cell in preparation for cell division. When we are talking about ordered sequence, we are referring to the simultaneous processes happening in the cell cycle. So cell cycle is represented by a pie chart like this, and it is composed of different stages, specifically four stage processes. So first is G1. So G1 or the GAP1, where cell increases in size, or there's a cell growth, followed by S stage or the synthesis stage, where DNA replication occurs. So DNA replication, we all know its main purpose is to produce two identical copies of the DNA molecule, which is essential for cell division during growth or repair of damaged tissues. And is followed by the G2, which has a similarities to the G1, which is cell growth, and also, but it is more on the preparation. It is a stage more on the preparation for cell division. And this three stage, the G1 as stage and G2 compromises the interphase stage. So the next phase is the mitotic phase, or where mitosis and cytokinesis belong, and we're expecting to have the formation, or to at the end of this process we're expecting two daughter cells. So here's the summary. We have the G1 as synthesis, the G2 and the M where cell division occur, and G1 where there is a cell growth. Now let's proceed to mitosis. So what is mitosis? Mitosis refers to the process of cell duplication, in which one cell divides into two genetically identical daughter cells. In the various stages of mitosis, the cell's chromosomes are copied and then distributed equally between the two nuclei of the daughter cells. So mitosis is a type of cell division, mostly done by body cells. And at the end of the mitosis, we're expecting to have two identical daughter cells. And we'll mention also about the distribution of chromosomes. So before mitosis, we have 46 chromosomes, and at the end of this process, the two daughter cells, the two identical daughter cells, will receive 46 chromosomes each. That's why we have equal distribution of chromosomes. So it's important also to take note about the responsibility of mitosis. So mitosis is responsible for the growth and repair. So mitosis is the reason why you are bigger and taller today than you are than when you are 5. And when you have wounds, the mitosis also is responsible for repairing those damage by cell division. If if cell didn't divide, you won't grow and there's no repair. So mitosis is very important and significant. Now let's proceed to the stages of mitosis. So let's proceed to the stages of mitosis. So here's the whole representation of the stages of mitosis. So the first stage of mitosis, prophase. Pro means before. So it's the beginning step. So you can see in here that the chromosomes are condensing. So it's the beginning nucleus is still there. And when I say condensing, the chromosomes are visible. The chromosomes are thickening. So that's the chromosomes, chromatids, and the centromere. So the next stage is the metaphase. M for middle. It's because the chromosomes will lined up in the middle of the cytoplasm, and the spindle fibers will connect to them, and it's begun to become visible, and it will connect to them, preparing for the next stage, which is anaphase. So anaphase, A for away, since the spindle fiber drags or move away the chromatids from moving towards the different pools. As you can see, they are moving away the chromatids towards the pools, and it's also preparing for the next stage, which is the telophase. So the telophase, each member of the sister chromatids reaches the opposite sides, and the spindle fiber here disappear, and nuclear envelope starts to reform. So, and the last step, the cytokinesis. So the cytokinesis is the ending stage where cytoplasm is start to separate and forming two daughter cells, and that's it. That's the two daughter cells. So here's the stages of mitosis. We have telophase, the cytokinesis, or the separation of the cytoplasm, prophase, the metaphase, or the middle, they're lining up, the anaphase, or they moving away towards the pool, and the telophase, and cytokinesis. So the products of mitosis, once again, we are expecting that mitosis will have, at the end of the process of mitosis, we have two identical daughter cells. Also, we have this representation, the table about the distribution of chromosomes. So during prophase, we have 46 chromosomes, but the chromatids is in double number, which is 92. Metaphase 46, and the chromatids 92 again. During anaphase, or the away, A for away, we have 92 chromosomes, but at the end of mitosis, we have 46 chromosome since the cytoplasm is already separated. That's why 92 divided by 2 equals 46. 46 chromosomes to each of the daughter cells. So now let's proceed to meiosis. So what is meiosis? Meiosis is a special type of cell division of germ cells and sexually reproducing organisms used to produce gametes. It involves two rounds of division that ultimately result in four cells. So when we say involve two rounds of division, it means that there's the two kinds of meiosis. We have meiosis 1 and meiosis 2, known as meiosis 2 as the equatorial meiosis. So meiosis is a special type of germ cells that we mentioned about sexually reproducing organism used to produce gametes. So we mentioned gametes, and gametes means the sperm cell and the egg cell, which is responsible for the reproduction, fertilization, and uh, forming of the new, creating new organism to reproduce. So the stages of meiosis. So as I said earlier, we have two kinds. We have two meiosis, meiosis 1 and meiosis 2. That's why we have a little bit longer process here. So let's proceed to meiosis 1 and with prophase 1. Pro means before again. So here in prophase 1, same with the prophase, in the mitosis, the chromosomes condense. So when we say condense, the chromosomes are thickening and visible, and the nuclear envelope breaks down and crossing over occurs. So next step is the prophase 1 stages. Before we proceed to the metaphase 1, we will pass first the different stages of prophase 1. So the first one is leptotin. So here the chromosomes thicken and become visible. So as you can see, chromosome starts to condense, thicken, and then visible. Next is zygote. So the homologous chromosomes enter synapses forming synaptonemal complex. So when we say synaptonemal complex, it refers to the two homologous chromosomes close together as you can see in the picture. We have the synaptonemal complex, or the homologous chromosomes close together. So this is the zygote. And the next one is the pachytene. So here, synapses is complete all throughout each pair of homologous chromosomes. So here in pachytene, the synapses is complete, but there is a very amazing process that is occurring here, which is the crossing over. So crossing over means genetic changes between non-sister chromatids of homologous pairs occurs. So as you can see, this is the crossing over, and after that we create this one, forming this one, and this is responsible for the variation. That's why in siblings they are not identical. That's why in the end of the meiosis we have unidentical and unique haploid daughter cells. It's one of the reasons the variation. It's because of the crossing over. So now let's proceed to the diplotin. So here, crossing over points as chiasmata and synaptonemal complex dissolves. Here's the chiasma, the resulting product of the crossing over, creating variation. Now, let's proceed to the diakinesis. Here, chromatids thicken and shorten, nuclear envelope breaks down, and spindle fiber begins to appear here. It's preparing to the metaphase 1. This stage, diakinesis is preparing for the metaphase 1. So now let's proceed to the metaphase. So metaphase 1, here pairs of homologous chromosomes move to the equator of the cell. Same with the metaphase and mitosis, they M for middle because the chromosomes are lining up in the middle, but the difference is here there's a pair, it's a pairing, they are lining up by pairs in the in the in the in the middle, and the spindle starts to spindle here by pair, and the spindle starts to connect them, preparing for the anaphase. A4 A4 away. So here homologous chromosomes move to the opposite poles of the cell. So the spindle fibers pull them down, move them to the poles, and this is this process preparing for the cell division, of course. So let's proceed to the telophase on of the last stage of the meiosis 1. Telophase 1 with cytokinesis. So chromosomes gather at the poles of the cells, the cytoplasm divides. So here we have two cells. The cytoplasm is the whole cytoplasm is separated into two with the chromosomes, of course. So this is the cleavage furrow. So now let's proceed to the meiosis 2 or the equatorial meiosis. So prophase 2, the same with the prophase 1 in the meiosis 1 and prophase in the mitosis. New spindle forms around the chromosomes. These are the spindles. And of course, the chromosomes are condensing and they are thickening and visible. And the different here is it's already two. It's already two cells. So now let's proceed to the 2. Metaphase 2 chromosomes line up at the equator. So it's the same with the metaphase in mitosis. The chromosomes are lining up in the middle, unlike in the metaphase 1, which is the chromosomes being pulled. There is a homologous chromosome pairing up in the middle. And let's proceed to the anaphase. Centromeres divide, chromatids move to the opposite poles of the cells. So in anaphase 1, the chromosome is being pulled by the spindle fibers, but here it's the chromatids. It's being moved away, it's being grabbed away from the middle, and it moves towards the different poles, pre preparing for the telophase stage. Telophase stage means 2 or 4. Here, the nuclear envelope forms around each set of chromosomes. The cytoplasm divides. So from 2, we have 4 daughter cells. 4 unique daughter cells. So the product of meiosis, we have 4 unique haploid cells. And this is the summary, the table representing the distribution of the chromosomes. We have at the prophase 1, 46 chromosomes and 92 chromatids. At the end of meiosis, we have 23 chromosomes and 46 chromatids. During phase 2, meiosis 2, at the prophase 2, the starting stage, we have 23 chromosomes from the meiosis 1. But the end of this meiosis 2, we have 23 and 23 chromatids. So now let's proceed to the comparison, the similarities and the differences of mitosis and meiosis. First, let's start with the differences. So mitosis, we have 4 stages in total plus interphase. So as we remember, have PMAT, the prophase, metaphase, anaphase, and the telophase, of course. And meiosis, we have 8 stages in total because we have meiosis 1 and meiosis 2, and it, every meiosis stage has 4 stages. So overall, it has 8 stages in total plus interphase. So happens in somatic cells. Somatic cells are any cells of the body except sperm and egg cell. So somatic cells are diploid. That's why we have in mitosis we have a 2 diploid daughter cells. And here in meiosis, it is happening in the germ cells, or the germ cells are biological cell that gives rise to the gametes of organisms that reproduce sexually in many animals. So the purpose of mitosis is cellular proliferation, or the cell grows and divides to produce 2 daughter cells, while the meiosis purpose is sexual reproduction, or to create another organism, to create another another human, another plants, another siblings, anything into sexual reproduction. So at the end of the mitosis, we have 2 diploid daughter cells, and in meiosis we have we have four haploid unidentical and unique daughter cells. So in mitosis also, with chromosome number remains the same. We already discussed it. We talk about 46 chromosomes at the start, and at the end we have also 46 chromosomes. But in meiosis, number is half in each daughter cell. And lastly, genetic variation doesn't change, and genetic variation increase. It's because of the law of segregation and law of separation, or the crossing over in the pachytene. It's the one of the reason of the genetic variation in meiosis. Here, and this is the comparison of the chromosomes. So in the in the mitosis we have 46 chromosomes at the end and 46 chromosomes at the starting point. And in meiosis, we have 46 chromosomes in the prophase 1, the starting of the meiosis 1, but at the meiosis 2, we have only 23. There's a decrease in distribution of the chromosomes. So here's the representation of the distribution of the microsomes, or chromosomes rather. Look at that. Meiosis, 46. The mitosis, 46 chromosomes, and the meiosis, 23. The N chromosome is 23. Now let's proceed to the eukaryotic life cycle, or the series of developmental stages. But we have three eukaryotic life cycle. We have gametic life cycle, the zygotic life cycle, and sporic life cycle. But I choose gametic life cycle. So gametic life cycle is reproductive found in animals and some protestants. The term gametic refers to the fact that gametes are the result of meiosis. So that's why I choose these cycles, because I can relate, because we experience gametic life cycle. We have the gametes, the egg cells, and the sperm cells creating a new human, new animals, new protestants, new plants. And here's the formation, the cycle, how gametic life cycle works. So during the gametic life cycle, as a reproductive cell produces haploid gametes, here, the productive cell produces haploid gametes. The sex cells, and such as egg cells and sperm cells that combine to produce a zygote, grows by cell division and cell lineation to produce multicellular. Here's the zygote to produce multicellular and diploid individual. That's us. In the gametic life cycle, the gametes are only haploid stage found in the life cycle, and the gametes are the only haploid cells produced. So that's how gametic life cycle is working. So that ends of my discussion for today about the cell cycle, and that could be all. And thank you for listening.